Okay, next I'll be discussing cell adhesion. There are five major classes of adhesion proteins. Adhesion proteins are integral proteins that mediate cell-to-cell binding. The five classes of adhesion proteins are integrins, cadherins, selectins, aminoglobulin superfamily, and mucins. We refer to these as cell adhesion molecules. CAM molecules mediate homophilic adhesion cells to a single type of heterophilic adhesion between two different cell types. Cell-to-cell adhesion involving cadherins and selectins depends on calcium ions, whereas interactions involving integrin and the Ig superfamily do not. Integrins mediate cell matrix interactions, whereas other CAMs participate in cell-to-cell adhesions as well. So in order for a cell's adhesions to function and integrate in a specialized junctions, they must be in a cluster of adhesion molecules. So there are four types of junctions: tight junctions, gap junctions, adherens, and we have in those areas you will have cell to cell and cellular matrix junctions as well. So first we're going to talk about the tight junction. The tight junctions are where epithelial cells prevent passage of fluid through the cell layers. In cell-to-cell and cell-to-cell matrix junctions, they also perform structural roles into holding the cell tissue together. And they carry out the role by connecting the internal cytoskeleton directly to the cell's exterior via another cell or through the extracellular matrix by cadherins or integrins. Actins and intermediate affiliates are connected to the cytoskeleton, to cadherins and integrins in the plasma membrane by adaptor proteins via tight junctions. In the cytoskeleton, associated junctions are organized by cell adhesion molecules, which connect the cell to another cell or to the extracellular matrix. And the adapter proteins, which connects the cell adhesion molecules to actin or keratin filaments and the bundle of cytokeratin filaments itself. And then there's the gap junctions. The gap junctions, cells and tissue directly communicate through gap junctions. Gap junctions are distributed along the lateral surfaces of adjacent cells and allow them to exchange small molecules. We also see gap junctions in neurons. Gap junctions, they're usually at the electrical synapse and allow ions to pass through one nerve cell to the next, and thereby allowing presynaptic cells to induce an action potential in the postsynaptic cell without a lagging period. And gap junctions that are in non-neuronal tissues help metabolic activity in all cells by allowing ion exchange of small molecules like cyclic AMP and DNA and RNA precursors. The next type of junction we have is the adherens. What we also call them is intermediate junctions or the belt desosome. This protein, you'll see them in protein complexes that occur in cell-to-cell junctions in epithelial and endothelial tissues. And how we would define adherens is cell junctions where the cytoplasmic face is linked to the active cytoskeleton. And we also refer to the adherens as also anchoring junctions because they hold cells together. And then adherents are basically a continuous bond of cadherents located on the surface before a tight junction, and they contain alpha and beta cadherents that link the E cadherents to the plasma membrane and a circumferential belt of actin myostatin filaments. They contain some proteins. Um, they contain the same proteins actually as focal adhesions, um, vinaclin, tryptomycin, and alpha actinium. Here's a diagram of basically the tight junctions, how these membrane interlock proteins together. I'm sorry, these are the tight junctions. And here are your intermediate filaments, the dust zones. These are the anchors that basically branch out, make sure they have support so cells won't break off. And the gap junctions uh, are these channels that allow, like you say, small molecules to pass through. So if we had a lot of calcium to flow through neurons, like in the you'll have this is how the action potential be generated by the influx of the ions. So now I'll get into the cadherins. The cadherins are calcium dependent family of cell adhesion molecules. They play a critical role in tissue differentiation, and cadherins are an integral membrane glycoprotein with about 720 to 750 amino acids. Uh, the cadherins also consist of an N-terminal extracellular region. The extra, the ex, ex, I'm sorry, the extracellular domain contains a sequence of sites necessary for calcium binding and cell adhesion. And during differentiation in some diseases, the amount of nature of the cell surface that can alter cadherin changes can affect cell adhesion and migration. Example of this will actually be cancer cells that have metastasized, like tumor cells. They usually correlate with the loss in cadherin function. That's how they can break off and go through the bloodstream and flow to different parts of the body, and they can metastasize in different regions. There are different types of cadherins as well. The E cadherin like holds epithelial sheets together, and removal of calcium, will disrupt the adhesion. And if the E cadherin's adhesion is blocked on aggregation, none of the cell specialized junctions between epithelial cells are generated. So, like I said before, and mentioned in cancer, if this particular one, something impedes its function, there will be no cell differentiation. So you have this continual growth of just general cell bodies, and they have no particular function. And these are also cadherins that you will see in different regions of the body. Of course, they have different specialized junctions for different regions as well. Next, we'll get into the Ig superfamily. One in particular is the NCAM. It's a nerve cell adhesion. This actually is a calcium independent cell to cell adhesion protein. The nerve cell adhesion molecule reflects the importance of nerve tissues. Just like cadherins, the NCAM mediates homophilic interactions, the binding to allow them to bind together. But unlike cadherins, NCAMs are encoded by a single gene, and the diversity is generated by alternative mRNA splicing and different and glycosylation. Some of the properties of NCAM is there are long chains of sialic acid, and they have negative charged sugars, so the carbohydrates on them are negatively charged. And they appear during the morphogenesis because they play an important role in the differentiation between muscle, glial, and nerve cells. Next, we'll talk about selectins. Selectins, also that you'll find in white blood cells. They participate in lymphocyte extravasation, extravasation, and it's required for the successive formation and breakage of cells to cells and content. Uh, Selectins are a class of cell adhesion molecules that are specific for glycoside vascular interactions. So they're really particular about just our immune system, the white blood cells in our immune system, and extra extravasation is is the movement of tissues. I'm sorry. And this is essential for monocytes, precursors of macrophages, which ingest foreign particles, neutrophils, which release several antibacterial proteins, T and B lymphocytes, which are antigens for the body. Then we also have P-selectin, which is localized by blood facing surface of endothelial cells. P-lectin P-selectin is a lectin. This is a protein that binds tightly to specific sugars. Uh, and as far as like tools and techniques, they use lectin is used to it has a general affinity for in chromatography to purify glycoproteins, or they use it as a reagent. Uh, Selectin is also calcium dependent. And in order for it to have these tight adhesions to occur, it must be activate occur. It has to be an activation between the endothelial cells and the leukocytes. The beta two containing integrins, and the integrins are another um, is another protein that's integrated between the cells. Then you have um, the binding of the PF. For here in the figure, you have the binding of the PF. You'll see this. These are the integrins, the alpha and the beta too. Uh, and this is the receptor and the T lymphocytes that's activated by this integrin in particular, the alpha L and the beta 2 through the Rho pathway. And integrins bind 1-cam and 2-cam, and I'm sorry, ICAM and ICAM2 and the IG superfamily. This is also a calcium independent signaling. So the IG superfamily actually has a variation of cell adhesion molecules, and some are calcium dependent and some are calcium independent. Integrins are a large family of heterodimer transmembrane proteins that promote adhesion of cells in the extracellular matrix or in the surface of cells. And last but not least, we just want to touch on mucins. They have a high glycosination, which they have a very high affinity to bind to sugars, and we see those in a lot of like mucus. They synthesis gel-like substances, so you'll see a lot of these bonds in our mucosal membrane, and that's the reason why they play a role in our primary defense for our immune system. Because when we sneeze and everything like that, like dust and dirt particles go through our nose, and a mucus catch those particles. So that's where you'll catch these bonds located. And here is just an overview of, some of the things that I went through. We can kind of see the cell-to-cell adhesions and the regions of the cell-to-cell matrix. So you'll have the actin filaments, the intermediate filaments, and they also labeled. So the actin filaments are coherent, and they'll have their bonds. And this is the belt that we were referring to, the adherent belt. Also, we refer to them as anchors. Desmosomes are also anchors as well. Then you have your tight junction here, where you see the membranes are really bound together so they won't break off. Then you have the gap junctions for the where the intermediate filaments are. I'm sorry, not the intermediate filament. These are connections, and this is where you have small gapes in between the membranes to allow for ion flow. Um, Here's another image of how some of the cell adhesion molecules, like the coherence overlap. The IG superfamily usually have like a bonding domain. Integrins also bound with each other. Like we said, homophilic. They bound with like integrins. And then selectins as well. It's like more like a lock and key type of adhesion. And for the cellular matrix, you have your integrins and your focal adhesions. These play a role in uh, the cell migration, especially focal adhesion. This allows for the elasticity for it to stretch and then generate this tension. So it's more like a rubber band. So when the cells want to migrate and they stretch, they generate this. They generate that force from that stretching, and then it protrude, and then they, kind of latch on, and then they kind of come right back.